Okay, and I think we're live. So I'm just gonna wait a minute or two for our late bloomers, and we'll get started today. Okay, I guess we'll get started. A lot of people running late today. It's kind of weird, but I got to get started because I got to actually finish this lecture on time today. I got to go straight to work. So we'll get started, and then as people want to join in, we'll get them to join in. Again, the live stream's going. Hopefully no one has any issues trying to attend the class today. Okay, so let's begin. So today the plan is to talk about a few things. We're going to talk about neuron structure. We're going to talk about the synapse. We're going to talk about glial cells. And we're going to be talking about the central nervous system versus the peripheral nervous system, as well as neural circuits. So we kind of touched upon this last year. We're going to get into a little more detail this year. And this is just to build on the foundational knowledge of what we talked about before of the three fundamental networks, which is the myofascial network, the circulatory, and today we're going to be going a little bit more depth into the neural networks. So neurons. These guys are the ones that are specialized for carrying signals from the brain, spinal cord to the rest of the human body, and then back from the rest of the human body to the spinal cord and back into the brain. So they operate both electrically and chemically, as we're going to kind of talk about in later. They connect at points between each other called synapses, and they target effector cells. So here is the structure of the neuron, and we're going to go piece by piece as to what each and every one of these things do. We have the whole structure here, and what we're going to do is we're going to kind of start from the dendrite, cell body, axon, hillock, all the way to the axon. Basically, the neuron is the basic working unit of the brain. This is where all the electrical signals are transmitted. And signals are transmitted to other nerve cells, muscles, and organs. It's also the neurons that sense changes in the environment and communicate these changes to other neurons. And so they command the body's reactions to these changes as well. So the very first component is the dendrites, and this is basically the extension of the cell body, which is this middle portion over here. This is the point at which chemical input is received from a neighboring neuron. And this is often the place that we call the starting point. Think of these guys as the antenna of the neuron. So this is where all the signals are being received. And again, this is basically the starting point of the nerve. Then we're into the cell body. Whenever you hear the term gray matter being thrown about the brain or the spinal cord, the pigmentation, that grayness actually comes from the cell body, so from this structure over here. Like any cell, the cell body contains organelles that are important to the neuron's own life and function, including things like the nucleus. So we got the nucleus over here. Think of this as the mini brain for the neural cell. And then we got mitochondria within the cell body too, which is like the energy source or the battery for the cell, producing things like ATP. And then other organelles in there as well. I won't get into too many details about them, but they're the ones kind of involved in regulating the production and transport of the cell's proteins. Um, this whole entire cell body over here is encased by a cell membrane that serves as a barrier between the inside and the outside of the neuron. And this is important as it helps maintain the neuron's electrochemical gradient, which is what we're going to talk about in a bit as well. But the electrical chemical gradient is basically the difference in concentration of certain ions like sodium and potassium, as well as the difference in the electrical charge between the inside of the cell and the outside of the cell. Okay. And just to kind of point out, Alzheimer's disease, for example, affects the neuron at the cell body. So things called tau protein, sorry guys, things like protein tau, they kind of accumulate as plaques in the cell body, and it ultimately leads to the degeneration of the cell. And that's where Alzheimer's kind of has its effect on the neuron. Oh, right over here, cell body. Okay. Moving on to the axon hillock. So the axon hillock makes the neuron particularly unique compared to other cells. It's the beginning region of what we call the axon, so that nice little wire over here. This is what a lot of people think of as the nerve wire. It's the last point of the nerve cell where all the charges are summed up. So we talk about at the dendrites of how all the chemical signals are received, and we're going to talk about more detail on that a little bit later when we get into the synapse. But these signals turn into electricity that runs through the cell and hits this point over here. Now, the charge has to reach a certain threshold over here. So there should be enough charge over here to hit a certain threshold. And if that threshold is hit, a signal called an action potential can kind of be propagated through the wire towards the rest of the nerve. Now, if the threshold is not achieved over here, the charge will not go down the cell, basically. Okay. Lastly, we're going to get into the axon terminal. So the axon is this long wire that extends from the axon hillock and carries electrical signals all the way down to the axon terminal. And these signals are called action potentials. I'm debating whether I want to get into the physiology of how these works, how these action potential works. It does get a little complicated. Now we're getting a little bit too microscopic, but I'll kind of think about it and I don't know, we'll see what the feedback is for that. But yeah, the axon carries that electrical charge that runs down all the way to the terminal of the nerve. Since it is a wire, insulation is of particular importance to the axon. And so you'll notice that it's wrapped continuously along the axon with a material known as myelin. This is a fatty membrane that's, wrapped around the wire over here, and it's preventing leakage of the charge. And it also insulates the charge, making it more effective, making it faster. And the signal kind of propagates all the way towards the end into the axon terminal over here. So the axon terminal is the site at which the axon comes into contact with other neurons and passes information to them. And we're going to get into that point over here called the synapse right now. Okay. So think of this as one of the branches that we just talked about over here. So this is the synapse is the contact point between the two neurons. And as you can see, there's no physical continuity between these 2 points, but there is a lot of exchange of information that occurs. And so the presynaptic terminal is analogous to that axon terminal that we talked about before. This is the incoming neurons axon terminal sending signals to the receiving neuron over here. This terminal over here stores a bunch of vesicles that contain chemicals called neurotransmitters. And these vesicles are stored in here and are basically waiting to be released. What triggers that release? It's that electrical signal that comes through the axon to the terminal that triggers the release of these vesicles. Okay, and so when that signal comes, the neurotransmitters are released into what we call this synaptic cleft over here, and all these little circles here are what we call the neurotransmitters. Again, this is just an artistic drawing. This is probably not how it looks, but this is the best idea of what we think it looks like. Okay. Now the postsynaptic terminal is the receiving cell. So this is analogous to, what do you guys think? What do you guys think this is? Based on all the structures we just talked about the nerve cell so far, what is the postsynaptic terminal? What structure is that of the nerve? So we know that the presynaptic terminal is the axon terminal, the end of the axon. What is the presynaptic terminal, guys? I'm waiting for responses in the comment section. Yep, that is correct. It is the dendrite. So this is the beginning of the mu nerve. Okay. Now, the reason we say that neurons communicate using electrochemical signals, electro meaning electrical, chemical obviously chemical, is because the signal inside the neuron begins as an action potential, which is an electrical signal. Now, this converts into a chemical signal the moment we're at the synapse, when the presynaptic nerve communicates to the next neuron using these neurotransmitters, which are chemicals. These neurotransmitters have to jump across the cleft and bind to receptors of this receiving dendrite or presynaptic terminal, which generates small changes through ion channels over here to generate the next electrical signal going down to the incoming neuron. Okay. So let's talk about glial cells now. Once again, I mean, we've talked about glial cells last year. These are 90% of the cells in the nervous system that get that really bad rep from the myth that we only use 10% of our brains. I'm a big fan of the movie Limitless, and they kind of quote that, and that's the only part of the movie that gets me. It's like, no, we use the whole entire brain. If we didn't, well, you're going to find out in a bit why that would be a problem. These are clearly very abundant in the nervous system with many different types, including the ones kind of shown in this diagram over here. They're responsible for the support of the neurons as well as maintaining ionic balances. So important ions like sodium, potassium, chlorine. They help with modulating neurotransmitter transmission. So they help support the transmission of signals down the neuronal cells. We know that through the myelin, of course, that are created by some of these glial cells. They help in the uptake and the removal of neurotransmitters from that synaptic cleft back to where they belong. They create scaffolds for the development of neurons or the recovery of neuronal injury. So they pretty much create this environment to allow the neurons to operate the way they should. They do not participate directly in synaptic interactions or signal propagation, but again, they do participate indirectly. And they assist in making these signals strong and efficient. So let's start first with my favorites. These are the astrocytes. So astrocytes are found primarily in the brain and the spinal cord. And they're actually the most numerous glia cell type we have. The reason being is because they're the space fillers of the neurons, and they provide a lot of the cushioning physical support. Um, because they're kind of so occupied almost everywhere, they also kind of control how much a cell can grow and retract within its own environment. Um, Now, apart from the physical support that these astrocytes play for the neurons, the most essential role of the astrocyte is the regulation of the chemical environment that surrounds the neurons, right? So they prevent things like leaking of a charge or any kind of neurotransmitters away from the synapse. They also assist in the reuptake of the neurons. So sorry, the neurotransmitters. So we'll go back to this previous slide over here. You'll see how all these neurotransmitters have to be released into the synaptic cleft, but eventually the signal will be transmitted, and these neurotransmitters have to go somewhere, right? Ideally, they should be going back into this presynaptic cell. And so the astrocytes will actually help assist in that reuptake of those neurotransmitters back into that presynaptic cell. Okay. Because these cells fill up most of the brain that isn't occupied with the neurons or the blood vessels, they're instrumental in contributing to what we know as the blood-brain barrier, right? This is a very tightly held together, heavily regulated barrier that helps protect the nervous system by controlling what goes in and out, right? So um, if we didn't have that control, anything could be influenced in the brain from our diet, from our environment, and that'd be a big no-no, right? Think of the blood-brain barrier as kind of like the border security we have nowadays in like the Canada or the States, and how really tightly controlled it is, and then multiply that by 1 million, and that's how stringent the blood-brain barrier is. And it has to be, right? You can't have any type of particle communicating to the brain. And that's what kind of makes pharmacology so difficult. So the development of drugs to influence the brain, you have to consider things like the blood-brain barrier. And the astrocytes are what are so instrumental in creating that barrier. Alrighty. Any questions so far, guys? Okay. Stop me if you guys have any questions, by the way. Again, it's kind of a little different today. We're going under the microscope, but I promise you we're going to be getting out of the microscope after too. Alrighty, let's talk about the microglia now. So these are the cells that are the scavengers. We call them macrophage-like. Think of them as cells that kind of scavenge around and look for things like debris or unwanted material that's kind of floating around within the nervous system environment. And so they kind of target them, right? These cells kind of move around, find these kind of different materials, engulf them like a macrophage will, and digest and disintegrate them so they don't influence the nervous system in a poor way. And so we can kind of call these cells the mini immune system for the neurons, because they modulate the inflammatory response to neurons. They're also instrumental in controlling the survival as well as the death of the neurons too. So they're kind of like the angels of death for the neurons. But obviously their support is instrumental in the fact that they protect the neurons from outside invaders, right? So we have the astrocytes, which function as the barrier. These are the guys that function as the, what's a good example? I guess the army that will kind of attack whatever incoming strangers make it through the barrier. Alrighty. Okay, last but certainly not least, we have what we call the myelinating glia. So over here, we've kind of circled the oligodendrocytes, but we also have what are known as Schwann cells. And we'll talk about the difference of the two in a minute. These glia provide layers of membrane we mentioned before called myelin that help insulate the axons of the nerve cell, kind of the same way the same way that plastic wraps around electrical wires, for example, and helps insulate electrical cords. Now, within these kind of wrappings, you're going to notice interruptions in that wrapping, and they have a specific name known as the nodes of Ranvier. I'm going to type that down because I don't have that labeled. Oh, let me send that to the rest of the group, not just one person. Nodes of Ranvier. And these are areas where the axon is exposed and where signals kind of jump through the electrical wire of the axon. So the importance of these is very, 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 very important. The importance is very important. How does that even sound like English? But one of the disorders that affect the myelin that I'm sure a lot of you are aware of is multiple sclerosis, right? So multiple sclerosis is a neurodegenerative disorder of the myelin sheath, the covering of these axons. And over time, what happens is there's a generalized degeneration of this myelin. And you can kind of imagine what happens when you lose myelin, when you lose that insulating cover of the axon, right? You have a loss of that insulation, which speeds up the charge. So over time, you'll have a slowing down of the charge that goes through the axon, and eventually you'll see that happen at a grand scale, which is like movement loss or the slowing of movement and the deterioration eventually, right? So there's obviously different types of multiple sclerosis. I'm not going to get into any details of it, but just know that this disorder kind of affects the myelin of the neurons, and that's the main reason why it affects it, right? It degenerates into the myelin, and the signal transmission is just not as efficient. Okay, so again, we have the oligodendrocytes here. They're found predominantly in the central nervous system, and so you can see there's a bunch of multiple processes. They can coil around several axons at the same time, and their nodes of Ranvier are more widely spaced. Schwann cells, on the other hand, I don't have a diagram. I should have created a diagram. But they're found in the peripheral nervous system. They're usually wrapped only around one cell at a time. And the nodes of Ranvier, those spaces in between, are much closer together. Okay. So let's get talking generally about the two different types of nervous systems we have and the subtypes. Again, this is just a refresher because I did mention I want to get into the nervous system a little bit just to give you guys an idea of what we're talking about. So we have the central nervous system as well as the peripheral nervous system, right? The central nervous system constitutes the brain and the spinal cord. And the body definitely considers the central nervous system to be extra important because both the brain covered by the cranium as well as the spinal cord covered by the vertebrae is heavily protected by a bony type of structure, right? And that's where the reason why is because most of the signal transmission has to come from the brain, and there are huge coils of neurons that are found in the spinal cord, and if you nick any of the two, much of the body can deteriorate because of that, right? Versus the nerves of the peripheral nervous system, they don't really get encased by anything bony. But if you nick one of them, there's not a huge physical consequence of that. Now, the central nervous system is pretty maintained in structure around most mammals. The brain's going to have most of its similar components, which is the cortex, the cerebellum, as well as the brainstem. So whether you look at rats, humans, apes, rabbits, we all kind of have the same structure to our brain, and the spinal cord is very similar too. Okay. What makes the human so unique is the angle of the brain to the spinal cord. So for us, we get a nice little 90-degree deflection, where the brain is oriented 90 degrees to the spinal cord. I mean, it will happen to the apes as well that are upright. But for most mammals, it's kind of like they're both along the same angle. So the brain is vertical, not horizontal. Okay. And now we got the peripheral nervous system. So I kind of, dimmed out the brain and the spinal cord here. I tried photoshopping these guys, these peripheral nerves, to stand out a bit better. Horrible Photoshop skills, by the way, but I tried. These are all the parts of the nervous system other than the brain, of course, and other than the spinal cord. We have two parts of the peripheral nervous system that help us function. So we have the somatic nervous system, which is the peripheral nervous system involved in anything that is under our voluntary or conscious control. So these are things like the motor nerves that exit the brain from the motor cortex, go down to all the separate muscles that we have. So if it's the bicep, causes contraction of the bicep to cause elbow flexion. If it's the quads, it causes contraction in the quads to cause knee extension. These are the nerves that we have conscious control over and that we use for movement. Okay, these are very important for our discussions going forward. We also have the autonomic nervous system. And this, on the other hand, consists of all the neurons that innervate things like the organs, the blood vessels, and the glands of our body. So we don't have much voluntary control, although I can argue we can somewhat indirectly voluntarily control these nerves. So we talked about last lecture how deep breathing can stimulate the vagus nerve. And that vagus nerve can activate to calm down things like our heart rate, right? So that's kind of the bridge between the voluntary and the involuntary peripheral nervous system that we have. But I mean, you'll notice the activation of the autonomic, the involuntary nervous system anytime you have an emotional reaction. For example, if you're getting really nervous, let's say, and you get that butterfly feeling in your stomach, it's because your autonomic nervous system is at play. You can't really control that feeling, it just happens. So your gut's secreting a bunch of different chemicals and your heart rate's starting to increase. You're getting a little sweaty in the hands, let's say, if you're getting a little nervous, your pupils start to dilate, ready to see anything. These are all things out of your control. And the body does these things to get you ready for whatever you're dealing with, right? And of course, the fight or flight response we know quite a bit about, as well as rest and digest, right? So that is what the peripheral nervous system is primarily about. Okay. So I threw this diagram here because I just wanted to highlight that although we can teach neurons in isolation, like we kind of did early in this lecture, they do in fact operate more like this, which is just this huge neural network, with trillions of potential connections, right? And this is the reason why we can have things like unlimited memory storage, right? I mean, what better memory device is there other than the brain? There's really no storage device out there that can store more information, right? I haven't hit a limitation how much I can remember. I'm pretty sure none of you hit that limitation either. So some of the main things I want you to consider when looking at this extravagant diagram is nerves never function in isolation. If something happens in the brain, it's usually a huge collection of neurons and nerves that must fire to create the experience we know of as movement and perception, right? And multiple parts of the brain are always firing. And when you go to sleep, they actually fire more. So our brain's kind of always working, right? And groups of neurons are the foundation of our sensation and perceptions, as well as how we respond to them. So this is just to highlight the complexity we have in the brain and how important it is not to just reduce to the level of the single neuron, right? So it's really important to talk about the single neuron, but remember, there are like billions of them. And it's kind of important to make that distinction as well. So that's the end of the lecture today. A nice, short, and sweet one. I made it to the half-an-hour mark finally, which is kind of cool. Just a note for next lecture, we're going to talk about the concept of tensegrity. So to kind of give you a bit of a background, tensegrity is the discussion of why we can, let's say, hurt our ankle and have neck pain as a result a couple years down the line. Our body is an interconnected network of myofascia, right? And so I want to explain why that's the case in the next lecture. So that's kind of like a little bit of a teaser of what we're going to do next time. The next lecture is going to be a little bit longer. So I hope not to get longer than 45 minutes because I don't know. I've kind of made the promise I'm not going to keep it too long, but that's the plan for next lecture, how everything is connected and why it's connected and how we can, address foot pain by not just looking at the foot, let's say, but looking at your knees, your hips, what's going on with your back. Um, and it's kind of like a big stab at the reductionist approach where a lot of therapists like physiotherapists like myself or chiropractors or whatever, when we look at injuries, a lot of us will think a shoulder injury requires shoulder intervention, but that's not the full picture, right? The body's so well interconnected, so interconnected, and to just address the shoulder would do it disjustice, would do it very little justice. So we're going to talk about tensegrity and how everything's connected. And I think that would be super, super interesting because it's a little more contemporary as well. A little bit complicated. I'm going to try to make it as easily digestible as possible. But yeah, that is the plan for the next lecture. So if you guys have any questions at all, throw me a comment right now, turn on the microphone, ask away. Again, this was just a little bit of a refresher for the nervous system, how the nerves work and how the support cells work. We're going to get back into the global scale now, so how the body functions next lecture. Bye, Deb. No worries. Hopefully you can fix that internet next time. Thanks for tuning in, though. Anytime, guys. Have an awesome week. Yeah, pretty heavy lecture next week, so we'll see you guys there. Okay, we have a question. Is myelin affected in Lou Gehrig's disease? I need a refresher in Lou Gehrig's disease. Let's see. It's ALS. It affects the nerve cells in the brain and spinal cord. Okay, so this is my very distant memory of how ALS works. Myelin is more of a peripheral nervous system problem, where ALS is more of a central nervous system problem, right? So it's still going to be affecting the myelin, from what I understand. I don't want to give you a bad answer though. Let me answer that next time for you, Melanie, because I don't want to do this poor justice. Yeah, you know what? Let me get back to you about that one. But I'm pretty sure myelin is implicated, and my best guess is that the difference between ALS and multiple sclerosis is ALS affects more of the central nervous system, whereas multiple sclerosis affects more of the peripheral nervous system. Okay, and yeah, no worries at all. Very important to know though, and yeah, that's my best stab at it for now. All right, guys. So if there aren't any more questions, I got to get jetting to work. But thank you all so much for tuning in. As always, it's always a pleasure. Next week, it's going to get back into, again, the human body, tensegrity. We're going to talk about why everything is so well interconnected and why you can't just fix a shoulder problem by addressing the shoulder, or you can't just fix a low back problem by looking directly at the low back. So hopefully you guys can tune in next week. I look forward to teaching that. In the meantime, peace out. We'll talk next week. Take care. Bye.